Okay, if there's no questions, I'm going to get started. Not hearing anything, so let's move on. Before I share my screen, let me mention that I've done, I think, all the grading for everything that had to be in before now. So the grade book is caught up. I do notice that it's not adding up all the points. And I'm assuming that it's out of 400 points. Is that what it says? Just a minute, let me check on that. Most of you, it's out of 400 points. And what I assume it's doing is there's some assignment that it's not counting yet because maybe it doesn't have to be turned in yet. I'm not sure. It's out of something like 440 points or 450 points total. And you can add all your points together and everything up and make a separate add if you want. I noticed that the percentage is pretty close to what it should be. Like I said, there's something like 450 points total so far. And for whatever reason, the grade book is only adding up 400 points. But most people have almost exactly the same percentage out of 400 as they would out of 450. And I'm assuming that the grade book, as the term goes on, it will add in the rest of the points. If that doesn't happen, meaning it's not adding in something, and I'm not sure why it's not doing that, I will have a separate total. I've already got it in here. Where is it? The total points out of 720. Of course, you can't see this. And then the percent. So I can put that in as a column. And that will be different maybe than the total that the grade book is doing. Now I'm hoping that they will be the same total, meaning that I think what's happening is the grade book's just not adding something in because you still have time maybe to work on it. And so that's why it's not adding it up. But I added it up separately. And like I said, it's out of something like 450 points. And all the points in the grade book are out of 400, with the exception of a few people. And I don't know why some people are different. Some people it's out of 415 points. But like I said, the percentage is basically correct for everybody. So hopefully you like your grade. Most of you are doing quite well. I noticed a number of people over 100%, and I said, oh, there must be a mistake there. Let me look this up. And I looked it up and it was correct, so that's good. Anyways, if you have any questions about the grade book, email me. If there's no questions about the grading, I'm going to begin the lesson. Like I said, almost everything is graded. And that's actually rare. I rarely do this. I get everything graded this early in the term. Let's see. Anything else I need to talk about the grading? I should state that on Thursday, December 2nd, I might be a little late getting the lecture started. I have a flu shot appointment on December 2nd. And I think that I will be home in time, no problem. But if I'm a little late getting the lecture started, please just bear with me. I shouldn't be late, but if I am, it'll just be a few minutes. So just warning you, the lecture might be a little late this Thursday. And I tried to get the flu shot. I tried all over to get the flu shot. And that's why I finally agreed to have it on Thursday, December 2nd, because I'm having trouble getting it. And I don't want to talk more about that. Let's talk about the schedule. So let me start sharing this now. Sharing the screen. Come on. I should be able to see the screen now. So today we're going to finish up the lesson, hopefully on chapter 13, viruses, viroids, and prions. On Thursday, we'll be starting chapter 7 and hopefully finishing chapter 7. In the lab today, there is nothing that we're doing. I will be there from 6:30 to 6:45 to answer questions. If you have any questions, log in at that time. There is no lab that we're doing. The unknown project is due Saturday at 11:59 p.m. The unknown notes has to be done and finalized by Friday. I think it's 11:59 p.m. on Friday. So if you haven't finalized your unknown notes, you should log into that page and make your last minute corrections by Friday, 11:59 p.m. Oh, there is something more about the grading I need to say. There were two people who thought that all the questions, let me see if I can find that. There were two people who logged into the last lecture assignment. Was that lecture 14 or lecture 13? I'm not sure which it was. Let me stop sharing my screen and I'll look into that. All right, you're not seeing the share. Anyways, they thought all the questions were answered. There are questions in part 2 of this lecture worksheet which have not been answered. So if you got a grade of 0.01, that means that there are questions in part 2 of the worksheet that need to be answered, and you will not get credit unless you answer a question from part 2 of the worksheet. So that is lecture 14 worksheet. No, lecture 13 worksheet, sorry. Yeah, there's 2 students who have a 0.01, and that means that they need to answer a question in part 2 of the worksheet if they want credit for worksheet lecture 13. I made it so that you can do that by Saturday. All right, let me shut the grade book and then start sharing the screen again. All right, if there's no questions about what we're doing, let's begin with chapter 13. So this was the last slide I was talking about last time, talking about the multiplication of animal viruses. Remember that the multiplication of animal viruses can happen in the cytoplasm, but it can also happen in the nucleoplasm. And mainly DNA viruses replicate in the nucleus of animal cells. The multiplication of animal viruses follows the basic pattern for bacteriophage multiplication, but there are some differences. Mainly, there's a difference in the penetration of animal viruses into the cells, and the penetration can happen by endocytosis or by fusion. Some older editions of the textbook, if you happen to have an older edition of the textbook, said that it happened by penocytosis. That's not quite correct. It's by endocytosis or by fusion. Fusion, which I'll talk about later, isn't shown on this slide. This is showing you endocytosis. where the virus attaches to the animal cell membrane, and then the cell membrane starts invaginating around the virus, endocytosis. And that's how it's brought into the cell attachment. The membrane starts invaginating. And then this pinches off here. So the cell membrane reforms. And then this is brought into the cell membrane by a vesicle. And there will be the cell membrane forming a membrane around the virus. So that's brought into the cell by a phagosome. The other way that the virus can be brought into the cell is for an envelope virus. The envelope of the virus can fuse to the cell membrane. And then the cell membrane, once it fuses to the virus, will go around the virus using the envelope as part of the cell membrane. And then this will open up, and then the virus will be able to move into the cell. Anyways, that's endocytosis and fusion. The uncoating happens by viral or host enzymes. Biosynthesis, that has to happen the same as for bacteriophages. The nucleic acid of the virus has to be synthesized, and then the viral proteins have to be made. And then the maturation happens very similar in bacteriophages, and the virus is assembled by the protein capsid coat coming together with the nucleic acid and maturing into the mature virions. The multiplication of animal viruses can be different than the multiplication of bacteriophages. The multiplication of animal viruses happens largely by rupture, and that happens with non-enveloped viruses. And this is a lytic life cycle for an animal cell. So the lytic life cycle can happen in bacteriophages, and it can happen in animal viruses. The animal viruses have no lysogenic life cycle, but they do have a life cycle that we call budding that is different. And a budding animal virus, the virus will replicate in the cell, and it actually will make proteins or glycoproteins that attach to the membrane, or well, we'll just say a membrane, cell membrane or some other membrane of the cell. And then the virus will come up to that membrane where the viral proteins are, and then push on that membrane and form a bud of that membrane. And then this bud will pinch off, and the part of the cell membrane having the viral proteins in it will then bud off and form the envelope of a virus. And then the cell membrane or the other membrane will reattach and reform. So this is how a budding virus obtains its envelope. And all enveloped viruses, at least animal viruses, occur by the budding life cycle, where the virus will bud out of the animal cell. A couple of things need to be stated here, and that is the virus just knows where the viral proteins are. And if this is a spiked virus, meaning that will be the glycoproteins that will be in the cell membrane, and the virus will come up to this region and then bud out of it, forming the envelope of the budding virus. This budding virus usually does not kill the cell. In fact, if the cell dies, it's not from the virus, It's because the host immune system recognizes this cell as infected by a virus, and then the host immune system kills this cell. The budding virus typically does not kill the cell. It buds at low enough numbers that it does not put a stress on the cell. Okay, any questions about that? Here we're seeing an actual cell in an electron microscopic image, and you can see buds on this cell, and that's from a budding virus budding out of the cell. Viruses have different genomes, as you know. There's double-stranded DNA viruses, single-stranded DNA viruses, double-stranded RNA viruses, and single-stranded RNA viruses. And so the virus will have to replicate its genome, and that will be different for each of the different viruses. The double-stranded DNA virus is just very easy how that one replicates. You separate the double-stranded DNA, and then you use one strand to replicate the corollary strand. Okay, anyways, that's how DNA viruses replicate. The RNA viruses replicate similar. The difference is that the single-strand DNA or the single-strand RNA will replicate by making the corollary strand. So we could say the positive strand RNA will make the negative strand, and then use that negative strand as a template to make the positive strand. So each virus needs to have a slightly different mechanism for the multiplication of its genome, depending on what its genome is, and then the biosynthesis of a virus. And you can see that there will be different mechanisms for DNA viruses and RNA viruses. And we actually separate the viral families into DNA viruses and RNA viruses. So let's first talk about the DNA viruses. Here we're looking at the DNA viruses, and there are 7 DNA viruses which infect human cells and cause disease. You don't have to memorize this table. Like the different family, I'm just trying to show you the 7 families of viruses and then the diseases they cause. And they're all DNA viruses, and it tells you the type of strand and whether it's an envelope or a naked virus, and then the size of the virus. It's useful information, but you don't need to memorize this table. You should just know that there are 7 DNA viruses. We've talked about some of them, like the pox viridae, the pox family of viruses, and the herpes viridae, the herpes viruses. DNA-containing animal viruses are the DNA viruses. DNA is generally copied in the nucleus of the cell. So that's a rule of thumb. DNA viruses are copied in the nucleus. The capsid and the proteins, however, are synthesized in the cytoplasm using the host cell's machinery for making the proteins. The multiplication of DNA viruses in a little more detail. Once a viral DNA enters the host nucleus, 2 things can happen. One is the viral DNA can recombine with the chromosome of the host cell and integrate into the host chromosome, cell's chromosome. In which case we do not call this a prophage because it's not a bacteriophage. It's an animal cell virus, so we call it a provirus, where the viral DNA integrates into the host cell's DNA. In a provirus, the viral DNA becomes permanently a part of the host cell's DNA. It is never excised. And this provirus will be replicated when the host cell DNA is replicated. Any questions about that? The other thing that can happen is that the viral DNA will be expressed, replicating itself, transcribing into viral messenger RNA, which will move into the cytoplasm to produce the capsid proteins. The capsid proteins will get together with the newly replicated viral DNA to form the new progeny virions. And then these virions will come out of the cell, depending on the life cycle of the virus. They could bud out of the cell, in which case the cell will remain alive, or the virus could lyse the cell in the lytic life cycle, and then that would release the new virions. So the multiplication of the DNA virus happens in this fashion. The DNA virus will attach to the cell membrane of an animal cell, and then it will come into the cell. That will happen by endocytosis or fusion of the viral membrane. Oh, and let me take a step back and talk a little bit more about the viral membrane. In the budding virus, I mentioned that that's how the virus obtains the envelope. The envelope is a part of the membrane of its host cell. And it rips off a part of the membrane to obtain that envelope. Okay, so you can see how the fusion of the viral envelope can usually happen with the cell membrane and then bring into the virus into the cell that way. Anyways, the virion enters the cell, it's DNA, so that DNA has to move into the nucleus before it can replicate, and that viral DNA will replicate to make new copies of the viral DNA. The viral DNA will also be transcribed to make viral messenger RNA, which will move into the cytoplasm to be translated into viral proteins, especially the capsid proteins, which need to get together with the viral DNA that's been replicated to make the new virions. And then these new virions will be released from the cell, either by budding, which is not shown, or by lysis of the cell, which will release the viruses. Any question on any of that? Okay. If not, that's it for the DNA animal viruses. Let's move on to the RNA animal viruses. Don't memorize this table. Let me see if I can blow it up. These are the number of RNA animal viruses, and I believe there's 12. 1, 2, 3, 15, sorry, didn't remember. There are 15 animal viruses in different families, 15 families of animal viruses that cause human disease. Most of them we've not talked about. We have talked about picornaviridae, but you probably don't know that term because I didn't introduce it, but that is the family of viruses that cause most human colds, the rhinoviruses, as well as the polio virus, it's a picornaviridae. I don't think we've talked about most of the other viruses, although we have talked about the coronavirus just in passing. We'll talk a little bit more about that, and we'll talk a little bit more about the retroviridae family of viruses. This is a RNA family of viruses that does not follow the rule of thumb. The retroviridae are an exception. All of the RNA viruses tend to replicate in the cytoplasm of the cell, with the exception of the family retroviridae. This is an RNA virus that replicates in the nucleus. And we'll talk a little bit more about that near the end of this lesson. Anyways, you don't need to know all of the RNA families or the strand type or the diseases they call. It's just this slide is showing you all of the viruses which cause human disease. Some of the RNA containing animal viruses, you'll notice that they have different sizes and shapes. And obviously cause different diseases. The replication of RNA viruses is similar to the DNA viruses, but obviously there are some differences because the RNA has to be replicated instead of the DNA genome. Viral replication happens generally in the cytoplasm. And remember, the positive strand RNA for single strand RNA makes the negative strand, and then it uses the negative strand as the template to make more of the positive strand RNA genome of the RNA virus. And the same with the negative strand genome RNA virus. It will make the positive strand and then use that positive strand to make more negative strand genome of the virus. So for the DNA viruses, it's very easy to replicate that, and that needs to be transcribed into viral messenger RNA, and then that viral messenger RNA needs to be translated into viral protein. But for the RNA viruses, we don't have this step. What happens is that the positive strand RNA virus, that genome itself can be read by the ribosome as messenger RNA and translated into the viral protein. How the positive strand RNA virus replicates is it makes the negative strand RNA and then uses this as a template to make more positive strand genome of the RNA virus. And yes, that positive strand can be used as messenger RNA. The negative strand RNA, to make more of its genome, will make the positive strand RNA and then use that as a template to make more of the genome, the negative strand RNA. And then it can also use the positive strand RNA as messenger RNA to make viral protein. Any questions about any of that? And then the newly replicated RNA gets together with the capsid proteins to make the progeny virions, the new virions. And these will come out of the cell either by budding or lysis, because we've only talked about those two life cycles for animal viruses, at least so far. Any questions about any of that? All right, so the positive single strand RNA virus, its virus will attach to the cell and then be brought into the cell either by endocytosis or fusion of the envelope of the virus to the cell membrane. It's an RNA virus, so generally that RNA virus will be replicated in the cytoplasm. If it's a positive strand, which is what we're talking about, that can also be used by the ribosomes to be translated into viral proteins. The positive strand will make the negative strand as a template to make more of the genomic positive strand. And then the genomic positive strand RNA virus will get together with the translated viral proteins to make the new virions. And these virions will then leave the cell either by budding or by lysis to release the new virions. Any question about that? For the negative antisense strand RNA, it's very similar. Everything's the same, except when the RNA is replicated, the negative strand will make a positive strand RNA. And then that positive strand will be used as a template to make more negative strand, the genome of the virus. And that positive strand can also be used as messenger RNA to be translated into new viral proteins, which will get together with the new negative strand genomic RNA to make the new virions, which will either bud out of the cell or lyse the cell so that the new virions will be released. Any question about any of that? Okay, if there's no questions, let's move on to the family of retroviruses, the family Retroviridae. This includes the lentiviruses, including HIV-1 and HIV-2, both of which cause human AIDS, as well as feline leukemia virus, which causes leukemia in cats and causes symptoms very similar to feline AIDS. This family is different because it's an RNA virus family member, but the replication of this RNA family happens in the nucleus instead of in the cytoplasm. What's going on is retroviruses contain the gene for the enzyme reverse transcriptase, and this enzyme can make DNA from RNA. So you take positive single strand RNA, which is the retroviruses, and in the presence of reverse transcriptase, I should have an arrow here, so we'll go. So I guess I have to add the reverse transcriptase here. Makes first single strand DNA, and then the reverse transcriptase takes the single strand DNA and makes double-stranded DNA. Any questions about that? This is one exception to the biological law that DNA always flows, or the information from DNA always flows from DNA to RNA to protein. Remember this? Put an arrow in there. In this case, the information is flowing from RNA to DNA with reverse transcriptase, and then from DNA, that information will flow to RNA as well, we'll just say positive single-strand RNA, which is the genomic RNA, as well as the messenger RNA, the viral messenger RNA. And so it can be used to make viral proteins. Which I guess I should have that as a D beforehand, because it's viral proteins. Which then gets together with the genomic RNA and the viral proteins to make the new virion. So in this case, information flows from RNA to DNA to RNA to protein, which is different from the information flowing from DNA to RNA to protein. This happens because of this enzyme reverse transcriptase, which is found in retroviruses. Oops, wrong one here. And actually, retro is from reverse, the RE, first two letters of retro, and then TR, the next two letters from reverse transcriptase, TR from transcriptase. And that's how retroviruses get their name, from reverse transcriptase. Any questions about that? Here we're seeing a retrovirus. You notice its envelope. It does have a protein coat. Inside, we do have a copy of positive single-strand DNA, and we do have a copy of the protein reverse transcriptase. I guess it's the pink one there. Yes, retroviruses tend to have two copies, which are both positive strand, single-strand RNA. You don't need to know that. So multiplication of retroviruses is a little different. The attachment, penetration, and coding of the retroviruses basically happens the same as animal viruses, which is similar to the what's going on with bacteriophages, but the biosynthesis stage is different. So RNA uses reverse transcriptase to first make single-strand DNA, and then the reverse transcriptase makes double-strand DNA. The double-strand DNA actually integrates into the host cell chromosome, which isn't shown here. And that double-strand DNA can be transcribed to make viral RNA, which is both the positive genomic RNA as well as the messenger viral RNA, which can be translated into the capsid proteins. The capsid proteins then get together with the newly made positive strand RNA, the genomic RNA, to form the progeny virions. Looking at in a little more detail, a retrovirus will bind to the cell. Retroviruses have an envelope, so they can have the envelope fuse to the cell membrane. The virus will then be dumped into the cytoplasm of the cell. The viral RNA will be reverse transcribed into viral DNA. The double-stranded DNA will then move into the nucleus and permanently integrate into the host cell's DNA. And it will permanently integrate into a chromosome of the host cell, the animal cell. And this we call a provirus. The viral DNA will replicate when the host cell replicates its DNA. That's not shown here, but I just thought I'd point that out. But more of importance to the virus is that the viral DNA can be transcribed to make viral RNA, which will be positive strand and can serve as both the new genome of a virus, as well as the viral messenger RNA. That RNA can be transcribed when it moves into the nucleus, excuse me, not the nucleus, the cytoplasm of the cell, into viral proteins, the capsid proteins, which will then get together with the newly replicated virion of the virus. And then it can most likely bud out of the cell. Retroviruses bud out of the cell. And that is how the retrovirus obtains its envelope, from budding out of the cell. That can be out of the cell membrane, out of the nuclear membrane, or out of the endoplasmic reticulum, depending on the species of budding virus. Any question about any of that? All right. Anyways, that's retroviruses and why they don't replicate in the cytoplasm is because retroviruses have a DNA intermediate which integrates into our host cell's DNA. Assuming this is a human cell, that would be our cell. All right, any questions about retroviruses? Oops, moving too far. If not, let's talk about coronaviruses. Most of you are familiar with coronaviruses because of our current pandemic of coronaviruses. But actually, coronaviruses is the name of the viral family. COVID-19 is the species name. The coronaviruses have actually been known to humans for a long time. They are single positive strand RNA viruses. They are enveloped, and they've been known for a long time because they are the second most common cause of the human cold. I mentioned before that coronaviruses are the most common cause of human colds. And I didn't mention adenoviruses are the third family of viruses that cause human colds. But coronaviruses have been known for a long time because they're the second most common cause of the human colds. And they cause a lot of colds, especially in children. Coronaviruses, besides causing COVID-19, also causes SARS, severe acute respiratory syndrome, and MERS, Middle East respiratory syndrome. SARS, MERS, and COVID-19 are all similar in that they cause respiratory disease, and they can all cause death. COVID-19, fortunately, I guess, causes less death than SARS and MERS. MERS, it has the highest mortality rate. About 36% of people who come down with MERS die. But fortunately, MERS does not spread from person to person. So if a person becomes infected with MERS, they do not spread that to another person. In fact, almost everyone who's come down with MERS have been in close contact with camels. Apparently, MERS is a disease in camels, and when this virus crosses over to humans, it has about a 36% mortality rate. So it's highly lethal. SARS is another lethal virus, and I think its mortality rate is estimated to be about 10%. And this is a disease that spread first in 2003. And fortunately, the health authorities were able to control that disease and they prevented it from spreading. And then SARS was no longer in the human population, but it came back at least once. I don't remember what year, and then started spreading again in China. Okay. And fortunately, the human health authorities were able to take control of this disease and kept it from spreading, which is a good thing because its mortality rate was around 10%. COVID-19 has an estimated mortality rate of between 0.2 to 3%, and it spreads more easily than the flu. One person is likely to infect 1 to 2.5 other people. And by the way, this information is before the Delta variant came out. I haven't updated this with the Delta variant. And my understanding is the newest variant of COVID-19, which I'm not sure how you pronounce this, Omicron and Omicron, something like that, is the newest variant. It has even a higher spreading rate than the Delta variant. So whatever their spreading rate is, it's higher than 1 to 2.5 other people. So if one person becomes infected with COVID-19, they're likely to infect 1 to 2.5 other people. And that's why COVID-19 has continued for this long. And that is, it's infecting at least one other person to 2.5, and then more people get infected, and then the disease continues. As I stated, the Delta variant and the newest variant are infecting even more people than this. And so COVID-19 spreads more easily than the flu. Hopefully you understand that. And you will take precautions to make sure you do not become infected. COVID-19 is the simple name of the virus. It's called COVID because that's from coronaviruses. And then the ID 19 comes because this virus was identified in 2019. It does have a scientific name, severe acute respiratory syndrome, coronavirus 2, or SARS CoV-2. However, the scientists do not even use this name most of the time because it's too similar to SARS, severe acute respiratory syndrome, coronavirus 1, I guess. And they tend to use COVID-19 as well. COVID-19 is closely related to bat coronaviruses and likely originated from the bat coronaviruses. When they sequence COVID-19, it's apparent that COVID-19 is actually a recombination of 2 bat coronaviruses and a pangolin coronavirus. so that two recombination events occurred with two bat coronaviruses recombining and a pangolin coronavirus. The pangolin coronavirus only contributes about 10% of the COVID-19 genome, and it is, what am I saying? It is part of the spikes of the virus. It's encoding the information in the virus for the spikes. So COVID-19 is something that probably happened relatively recently, probably in bats, and that's why we've never seen it before. And they believe that this virus was being transmitted in bats and likely was transmitted to an intermediate animal, such as the pangolin, before it was introduced into humans. There may have been only one pangolin that introduced it into humans. We'll talk a little bit about that. And that's why we've never found the intermediate. To my knowledge, we've actually never found COVID-19 in bats yet either. And anyways, this is the belief, but it does not prove that this is what happened. But it's likely that COVID-19 was a recombination of two bat coronaviruses and a pangolin virus. And then it somehow got introduced in humans and then spread very easily among humans. And then the virus is now in humans. Any questions about any of that? All right. COVID-19 is primarily spread via droplets from colds and coughs and sneezes within a range of about 6 feet. You should know it can spread by other means, meaning you could be further than 6 feet away and you could get the infection. It may spread by indirect contact, meaning a contaminated surface may pick up the virus, and then you may pick it up, especially on your hands, and then you inoculate your a mucous membrane in your face. As I mentioned, this is the primary way the colds and flus tend to spread. But for whatever reason, COVID-19 prefers to spread directly from person to person. And you have to be within a range of 6 feet for the virus to move from one person to another person. And it can spread simply from the person breathing without a mask on or the person talking to you. It doesn't have to be a cold or a sneeze. So it's likely breathed in, but it can be picked up by people's hands and then carried to the face. In fact, they do know that COVID-19 can survive for up to 3 days on plastic or some metal surfaces, but on paper and cardboard, COVID-19 will die after a day. So on paper and cardboard, the virus dies sooner. Okay? So yes, you could pick up COVID-19 from the mail. And what I do is I just leave my mail out for a day. And then the paper of the mail, the virus will die. And as I stated, COVID-19 doesn't like to spread that way for whatever reason. It tends to spread directly from person to person. And that's good for those of you who go grocery shopping, because perishables, you pick them up from the store You don't know somebody who had COVID-19 was touching that perishable before you, and then you take it home and you put it in your refrigerator. Well, the COVID-19 will survive longer in the refrigerator than it will at room temperature. And I'm not going to wait for a week for the perishable before I use it, because something like milk, for example, will tend to spoil pretty much in about a week. Actually, most perishables will tend to spoil in a week's time. But fortunately, COVID-19 doesn't tend to spread that way. You could wash off the perishable if you wanted to. COVID-19 symptoms, they're mostly flu-like symptoms, though actually runny noses and sneezing are less common than you have in the flu. And then the shortness of breath and the deaths are more common than we see in the flu. The mortality rate with COVID-19 increases with age of the patient, with the ill health of the patient. Deaths are more common in men. And for whatever reason, deaths are also more common in Blacks and Hispanics than they are in Asians and whites. And what's going on there has never been explained as to why that is the case. It's pretty obvious that people who are older are more susceptible, people who have ill health were more susceptible to death. What isn't quite ever explained is usually most viruses tend to be worse with the young and the elderly. And COVID-19 does not tend to hit the young. Okay, and that's just the way this virus is. It's sparing the young and hitting the elderly. The incubation period from when you're first exposed to the virus to when you have the first signs and symptoms range from 2 to 14 days. You should realize that some people are asymptomatic, so they will never have signs or symptoms of COVID-19. And it's estimated by the CDC that about a third of Americans will be asymptomatic for COVID-19. So a third of Americans will have COVID-19, but they will be asymptomatic. So you will never know that they have COVID-19 and could spread it to you, meaning that these asymptomatic people are capable of spreading COVID-19 around. And most of you are fairly young. I will state that for young people, the number of asymptomatics is about 50%, meaning it actually increases for children and young adults because the virus is just less severe in them. And so more of them will be asymptomatic. The estimated death rate is between 0.2% and 3%. The actual death rate in the United States, admittedly, this is probably about 6 months old now, it was about 1.8% of those people getting infected with COVID-19 were dying in the United States. And if you look at different countries, it'll be different. In Mexico, it's much higher than it is in the United States. So the death rate, the last I saw, was close to about 10%. Now, admittedly, in Mexico, they're not sampling as many people. So there's probably a lot of people who have been exposed to COVID-19 who didn't know they got COVID-19 infection. And so there's probably that, as well as the fact that Mexicans are Hispanic, and so they're going to have a higher death rate than in the United States because we don't have as many Hispanics as Mexico has. All right. Any question about any of that? Anyways, the death rate depends on the country, but the total death rate is estimated to be about between 0.2% and 3%. And you know, now that I think about it, that is probably an estimate before the Delta variant. And the Delta variant is more severe. So that number could be different. It could be higher, but it could be lower. Who knows? I have to look up. As I stated, these numbers were actual numbers from 6 months ago. Prevention, how you can prevent getting COVID-19 is to use correct hand washing. When you wash your hands, you should wash them with soap and water for at least 20 seconds. Keep your distance from people. Stay 6 feet away if you can. Avoid people who are coughing. Avoid touching your face. And then wearing a mask, especially when you're in public. And of course, the best way to prevent getting infected is to get the COVID-19 vaccine. If you're sick, you should wear a mask all the time, especially when you're in public. It's best to stay home and to cough into your elbow or tissue. And of course, wash your hands frequently. Any question about that? All right, that's it for COVID-19. If you have any questions, you can look at the CDC website or send me questions. Like I said, my data, I've got it actually here, this is the last information from, what is that, May of 29, 2021. All right, let's move on to viruses and cancer. Viruses can cause human cancer, and I should state that that's actually a correlation. What's going on exactly is a little bit more complicated, and we'll talk a little bit about that, how viruses are causing human cancer. And about 10 to 15% of all human cancers are started by a virus. Like human papillomavirus can cause cervical and skin cancer. Hepatitis B virus and hepatitis C virus are well known to cause liver cancer. A recent study by Tang in 2013 found that not all cancers are started by viruses, like breast cancer and glioblastoma. They're not caused by human viruses, but some viruses are. Cancer is uncontrolled cellular growth. We're not going to look into that. Has certain properties. How viruses cause human cancer are in two ways. The first way is that the virus can form a provirus, meaning if the viral DNA integrates into our cells' DNA, that can muck up our DNA. The virus permanently becomes the genetic part of our cell, meaning the viral DNA integrates into our host cell DNA. When that happens, that can muck up the DNA in the region around the virus. The viral DNA can integrate into a gene and obviously foul up that gene, so that gene is not turned on. It can mutate into a region of the DNA where that region is turned off, and then the viral DNA can turn on the cell's DNA. Anyways, the point is that this viral integration can mutate our DNA, and then that can start the cause of cancer. Any questions about that? That one's pretty obvious. There are also oncogenic viruses. An oncogenic virus is a virus that contains a gene that we call an oncogene. An oncogene is a mutated form of a normal cellular gene involved in cell growth. So this gene, in its normal state, helps the cell grow. Well, the mutated form of this gene causes the cell to grow and grow and grow and transforms the normal cell into a cancerous cell. Is that clear? So it's a mutated normal gene involved in the cellular growth of the cell, but it's mutated, and so it causes the cell to grow abnormally, which can start the cell into cancer. Any question about that? Some oncogenes, the oncogene can actually be integrated into the host cell's DNA. That's not terribly important. The important part is that this oncogenic virus has as an oncogene, and then when this gene is expressed, it leads the cell to grow. And uncontrolled growth is the start of cancer. All right. So we've talked about, oops, wrong one. We've talked about lytic animal viruses and budding animal viruses. Animal viruses have a few other life cycles. There's also the latent life cycle. An animal virus that goes into the latent life cycle is an animal virus that remains, will enter a latent life cycle, and the virus will remain asymptomatic as long as it's in the latent life cycle. The host cell can go latent for long periods, and then the cell can come out of the latent life cycle and then can enter a active life cycle where the virus is actively replicating. That's usually a budding virus that can enter the latent life cycle, but it could be a lytic life cycle as well. Cold sores is an example of a virus that when you have the cold sore, the virus is actively replicating by the budding life cycle, but the virus can enter the latent life cycle, and then this patient has no signs or symptoms of disease. The virus is not actively replicating, it's just hiding out in the cell, and then it can become active again, causing another cold sore, and then the virus is left the latent life cycle and enters the budding life cycle. The best example of this latent life cycle is when a child comes down with chickenpox. The virus is actively replicating in the budding life cycle, and then the child's chickenpox goes away. That's because the virus has entered the latent life cycle. And then when the virus leaves the latent life cycle, the patient comes down with shingles. And this is usually when the patient is in old age. Okay, so the virus has gone latent for decades. There's usually a trigger for disease manifestation, switching the virus from the latent life cycle to the active life cycle. There's also the persistent viral infection. This is a disease process that occurs over a long period of time. Usually the virus starts very low in number and can be detected, but over time, and it could be a long period of time, the virus numbers increase. The virus is never cleared by the host immune system. In some cases, it may be fatal, like subacute sclerosing panencephalitis. This is caused by the measles virus. And then HIV causing AIDS can be fatal. In the early days of an HIV infection leading to AIDS, it was almost always fatal. Now they have medication to keep the patients alive. But the point is HIV can be fatal, and it is a persistent viral infection. There are persistent viral infections where the patient can tolerate the persistent viral infection. This happens with cytomegalovirus, where about 50% of Americans comes down with this virus, and they just tolerate it, and they have a persistent viral infection. Unfortunately, cytomegalovirus is tolerated. It is not ever building up to levels and becoming fatal. Another example is varicella zoster. The patient has chickenpox, and then the next time it comes out, it causes shingles. And then the shingles in these patients continue for the rest of their life. This is very rare. Less than 1 in 1,000 patients who have shingles comes down with this. But what happens is the virus enters the persistent viral infection and it's tolerated. You have shingles for the rest of your life. There are a few other herpes viruses which become persistent viral infection, and they're tolerated for the patient's life. So normally with most viruses, we have an acute infection like a cold or the flu. The patient has no infection. They're exposed to the virus. And then a short after, the virus can be detected and the patient has acute infections, a large number of virions, and then the virus goes away. It's cleared from the patient by usually their immune system. It goes away. The patient has no symptoms, signs or symptoms, and the virus is gone. Okay? In many cases, it doesn't come back, like H1N1. We had that, and you might have gotten infected. You don't tend to get it H1N1 multiple times where it comes back. Although with a latent infection, a child gets chickenpox, they have the acute infection, they have signs and symptoms of chickenpox, and then the latent infection, they have shingles, but that usually goes away, although the patient could have shingles again and then comes back and have another latent infection. But anyways, that's a latent infection where for many, at least with shingles, for many years, the patient had no signs or symptoms, no virus replicating. Okay, and then it comes out. Cold sore is another example. The persistent infection, the patient gets exposed to the virus number builds up over time, and then they have a bloom, much worse signs and symptoms. And you see how that's broken? That's because the patient dies. That doesn't have to happen in a persistent infection. It could be that the virus is just tolerated, and so you have high numbers of the virus being shedded by the patient until the end of their lifespan. Prions, let's briefly talk about this. Prions are infectious proteins. Prions comes from proteinaceous infectious particles. They are inherited and transmitted by ingestion, transplant, and surgical instruments of the infectious proteins. These proteins cause spongiform encephalopathies, vacuoles in the brain. which you can see in humans and other animals. Animals, we call it bovine bovine encephalopathy, more commonly called mad cow disease. There's also sheepies, sheep scrapies, severe chronic wasting disease, which we won't talk about. In humans, we don't call it that. We usually call it Creutzfeldt-Jakob disease or Gerstmann-Straussler-Schneckner syndrome, fatal familial insomnia, and Kuru. Let's talk briefly about Kuru. This was a prion disease seen in the Fore tribe of Papua New Guinea, shown here where the Fore tribe is, where the patients were unable to walk, lost the ability to swallow or chew, drastic weight loss, and eventually led to death. How it was transmitted? The Fore tribe had an unusual funeral ritual that involved family members cooking and eating their dead relatives. What happened was, prion happened in the Fore tribe. Somebody died of prion disease. The Fore tribe cooked the individual and then ate the dead, especially the nervous tissue like the brain. And then the prion disease was spread in the Fore tribe as Kuru. Spongiform encephalopathy. Here you see a picture of a normal brain, and there's a Kuru brain, a classic CJD disease, scrapies, where you can see that the brain has the appearance of a sponge. Encephalopathy means sponge. Prion is a proteinaceous infectious particle. The mechanism of how the disease spreads, how the prion replicates itself, is there's two forms of a protein. And the normal form is PRP hyper C, meaning subscript C. That's the normal protein, which is made in the nerve cell. It's needed by the nerve cell to function. So it's made by the nerve cell. And there's the prion form of the protein, which is PRP-SC for scrapies. And what happens is the PRP-SC protein comes up to the PRP-C protein and then causes the normal protein to fold in its protein folding and become the PRP-SC protein. How is that possible? The PRP-C protein and the PRP-SC protein have the same amino acids. They're the same protein, they just have a different folding pattern. And the PRP-SC can fold the normal protein into the scrapies protein. The cell now has two scrapies protein, but not enough normal protein. So the cell makes more normal protein, but now there's two scrapies protein. And if one comes to the normal protein, it then changes its folding pattern into the scrapies protein. So the scrapies protein accumulates in the nerve cells, forming the plaques in the brain cells that we saw earlier. This will lead eventually to cell death as the scrapies protein accumulates into the cell, and the scrapies protein just accumulates more and more into the cell and eventually kills the cell. All right, mad cow disease was a problem that happened in England and Britain because the British had the I don't know, insane habit of wanting to use the cow brain, which nobody wanted, didn't want to eat cow brains. And so they ground up the cow brains and then fed it or put it on grain and then fed that grain to other cows. And as the cows were eating cow brains, you can see how the scrapies form of the protein got into the cows and then made more scrapies protein, causing mad cow disease in England. All right, any questions about any of that? If not, let's briefly talk about plant viruses and viroids. Plant viruses resemble animal viruses in many ways. The difference is that plant viruses have to go through tough cell walls. So plant viruses tend to enter plant cells through wounds in the plant via parasites like nematodes or fungus or insects that enter the plant, causing wounds in the cell, then allowing the virus to enter the plant cell. Plant viruses are important for causing many diseases of economically important crops, such as beans, corn, sugar cane, potatoes. Infected plant cells can spread the viruses in pollen and in their seeds. Any question about plant viruses? If not, that's all I want to say, because plant viruses do not cause disease in animal cells. So that's all we're going to talk about then. Let's move on to viroids. Viroids are something that causes disease in plant cells. And what a viroid is a naked RNA which causes disease in plant cells. So think of it as a virus, a naked virus, without the protein coat. And then this RNA molecule gets into the plant cell and then causes disease. It does replicate in the plant cell. Fortunately, there are no viroids known in animal cells. So viroids only cause disease in plant cells. One of the diseases they cause is potato, the spindle tuber viroid, which causes the potato to grow smaller potatoes than the plant normally would. Any question about plant viroids? All right, if that's it, I'm going to say that's it. That's all we're going to talk about. I will see you, at 6:30 if you have questions for the lab. Bye.